Buenas tardes, bienvenidos a las charlas de la Conavio. Como ustedes saben, estamos en nuestra séptima charla del ciclo Los ecólogos de Krebs sobre regulación de poblaciones. Y este jueves tenemos otra vez la presencia de la doctora Judy Myers, profesora emérita de la Universidad de Columbia Británica, quien nos va a hablar regresamos a los insectos, nos va a hablar de los ciclos de 10 años en insectos, principalmente orugas de polillas que causan grandes problemas a los bosques templados. También va a hablar un poco de la mariposa monarca, lo cual es también, muchas gracias, es muy interesante haber metido ese tema. Y como les comenté, tenemos una página web que hemos desarrollado, que verán ahí en el Facebook, en donde pueden descargar las presentaciones, ya están traducidas al español, y estamos también subiendo las presentaciones por ahora, los videos en inglés, pero también serán traducidos. La doctora Judy Myers es ecóloga canadiense y americana, y como muchos de ustedes ya conocen, se ha dedicado a varios temas de ecología aplicada, en donde la ciencia de la ecología de poblaciones es crítica. Ella ha trabajado con patógenos de insectos, con control biológico de insectos y de plantas, entre otros temas. Es profesora emérita en el Departamento de Zoología de la Universidad de Columbia Británica en Vancouver, Canadá. Y yo tuve la gran oportunidad de conocerla mientras estuve en Vancouver haciendo la maestría y más tarde el doctorado. Ella ha trabajado y ha obtenido muchísimos reconocimientos, tiene cientos de publicaciones, y pueden visitar su sitio web a través de esta página que hemos colocado. Tenemos un enlace directo a su sitio web. Muchísimas gracias a Judy por aceptar ser parte de este seminario, y vamos a presentarla en inglés. Thank you everyone for being here. We are on our 7th seminars of the series Krebs Ecologists on Population Regulation. And today we are back to the insect world, and Judy Myers is going to talk about nature's 10-year cycles, the cyclic dynamics of forest caterpillars. She's talking mostly about insect species in northern forest, but she also included, and thank you very much for that, a little bit on monarch butterflies, which everyone is familiar here in Mexico. As I said last time, Judy Myers is a Canadian-American ecologist, and she's done research on a number of topics of applied ecology, where the theme of population ecology is really critical. She's worked on pest outbreaks, insect pathogens, and biological control both of insects and weeds. She is Professor Emerita in the Department of Zoology at the University of British Columbia in Vancouver, Canada. And many years ago, when I was a student of Charlie Krebs in Vancouver, I had the pleasure to meet Judy, to spend time discussing as a student many publications. And recently I learned that she also worked on monarch butterflies, and I guess that's why she's presenting us some of that information today. Thank you, Judy, for that. She has received many awards. She has hundreds of publications, and you can visit her website through the website that we developed for these seminars. In our website, which will be posted on the Facebook, you can link to her site. You can also download the presentation in Spanish and in English, and we will put in there also the video of this presentation today. And well, thank you, Judy, for being here again today and talking about a different topic, a different related topic, which is nature's 10-year cycles, the cyclic dynamic of forest caterpillars.
Thank you, Carlos.
You're welcome. There you are. That's great.
Everything
Perfect.
fine? Good.
Yeah, this is great.
It's very nice to be here and to have this opportunity to talk about a couple of my favorite things. One is the cyclic forest caterpillars, and the second is monarch butterflies. We've, in this series, talked about other population cycles, the hare cycles. Stan Booten told us about how the hares and the lynx are intertwined in the population cycles of the snowshoe hare and the Yukon, and also how there are components of changes in reproduction with the population cycles, which changes the number of generations over a summer, as well as stress to the populations when they're at high density. And we talked about the voles, and Charlie Krebs told us about the 3-year cycles, and stressed, I think, that the importance of social interactions. It's more than just numbers, more than just predators, but social interactions amongst the voles that determine the cycles. What I'm going to talk about today are some examples of population cycles of forest caterpillars. And I have here on this graph my favorite, the 10 caterpillars in the black symbols. And I've also plotted on here is another cyclic species, the autumnal moth, in the red triangles. This is work that's gone on in Europe, and we'll talk more about both of these species in a moment. Here are some examples of the best studied of the forest caterpillar cycles. In Switzerland, we had the larch bud moth. In Sweden, Finland, and Norway, the winter moth and autumnal moth. 10 caterpillars in North America, Spruce Budworm in North America, and Gypsy moth, which was introduced from Europe to North America. And I'm going to discuss some of the details about these different species. What is required for cyclic dynamics? Well, first of all, the ability to increase very rapidly, to have a high reproductive rate. And to this end, many of the species that go through these population cycles are what we call capital breeders. That is, that the moths don't feed after they come out because they have all their eggs already developed, and they very quickly lay those eggs. The other thing about these forest caterpillars is that their food is widespread. The forests are made up of often just a few trees, and so there's a huge food supply for them. The other thing is that each of these species has a very strong mortality agent, and those are primarily diseases or parasites. And then there is a delayed mechanism that is required to prevent an immediate recovery once the population is declined, and this is referred to as a second-order feedback mechanism. I'm going to talk about the patterns of density change in forest caterpillars, what causes the population decline, and that will refer to parasitoids and diseases, and also synchrony among populations, which I find a fascinating phenomena. And it requires dispersal to be occurring, and we will look at genetic similarity that will be related to this dispersal. Finally, I will mention an example of a non-cyclic species, the monarch butterflies. Starting with the larch bud moth, this is a picture of their habitat in the Swiss Alps. You can see the little caterpillar there on larch. And one of the things about larch is that it's a deciduous tree, even though it has needles. So it loses its needles in the winter and develops them again in the spring. This is a picture of Werner Baltensweiler, who studied these larch bud moth over his whole career, an amazing man. And this is data from his studies that started in the mid-1940s and continued until 1990. What we see here is the increase phase of these population cycles takes about 5 years, and then the decline phase occurs over approximately 4 years. You'll note that there is no period of low numbers. This, you will also notice, is plotted on a logarithmic scale. And this maybe gives you this more realistic impression that the populations go up and down without long periods of low numbers. What Dr. Baltensweiler did was counted larval density by looking at the number of those little caterpillars per twig. He monitored what proportion of those larvae were parasitized, and also measured leaf length, because there was quite a bit of interest in the impact of these caterpillars on the food supply. Defoliation by the caterpillars reduces the length of the leaves the next year, and they monitored this over time. And also it causes those leaves to be of poorer quality, and this feeds back to the moths so that they have a reduced fecundity. Here is a graph that comes out of the review that I did with my colleague Jenny Corey. It's published in 2013. And the blue areas represent the population density of the large bud moth. The red dotted lines are the parasitized larvae, the proportion of caterpillars that were parasitized. And then the blue is the leaf length that they measure over time. And we see here a clear pattern of parasitization occurring late as the population is going down, and the leaf also changing with the population density, being long during the increase phase, and then it drops down after the peak of density. So we have two factors going here. One is parasitization, and the other is changes in food quality. Now, Turchin and others in 2003 did a model of these populations, and I have put the model here for you to have a look at, but it's not really that important other than it contains the density and the parasitization. And we see quite a good correspondence between the model population in the open circles and the actual data. Turchin and colleagues concluded that we didn't need anything more than to understand this relationship. It was all parasites. But what about the leaf length and its impact on the fecundity of the moths? Well, their summary was that a simple model of bud moth parasitoid interaction accounts for 90% of the variance in the population dynamics. And plant quality on top of that has just a very small effect. And we see here they added a factor for leaf length in there. However, it's interesting that Baltenswider, much before that, reported that fecundity of these moths declines by 75% to 85% in the year following this defoliation, here and here. So it's hard to believe that this isn't having an impact and isn't delaying the recovery of the population, because in the models, fecundity wasn't modeled directly. It was the rate of parasitization and a leaf length factor. So I still think that the answer is still out on that. The fecundity could be involved. So it remains a question then if variation in fecundity is a contributing factor in the cyclic dynamics of larch moth, bud moth. Now the next species I want to talk about is also from Europe, and that's the autumnal moth, Eperita autumnata. You see here on the left a defoliated birch forest in northwestern Europe, or the Scandies, and this is the impact of these moths on the foliage and on the trees after a period of high density. Olo Tenau, again, much like Baltenswider, studied these moths for years and years and has put together quite an impressive data set. More recently, Tero Klemila has followed up. And in fact, in Sweden, Finland, and Norway, there are many researchers who have contributed in ways that I cannot totally cover today to the understanding of these dynamics. Again, we have a picture here. Within the blue is the density of the autumnal moth, and in the red is the rate of parasitization, the proportion of larvae that were parasitized. And we see as the population goes down, or when parasitization is high, the population of the host, the autumnal moth, drops. And we get quite a consistent relationship there between decline in the moth population and high parasitism. Well, it's always nice to have an experiment to further elucidate these factors. And Nata Kremla did an experiment and published in 2010 in which she excluded parasites such as this wasp here from 4x4 meter cages, and she had cages that had holes in them so the parasites could get in, and cages that were totally enclosed. Now what's happening here in the population at this time is you can see the population has declined to very low density while parasitism continues relatively high. And in this exclusion here, the autumnal moth is able to take off again immediately, whereas the controls the population stayed down because they are being exposed to this parasitism rate that we see over here in the surrounding populations. So this is a good indication then that parasitism does have a major factor on the population density of autumnal moth. However, again, there's another factor, and that's moth size. And in insects, of course, size is closely related to fecundity. And we see that when the density gets really high, the size of the moths, and they measured the leg length, and the size of the moth declines, and then the population goes down and the moths get bigger again. So there's a direct correspondence between the size and fecundity of the moths and what the population dynamics are doing. So what is driving the population dynamics? The reduced fecundity at high density, high parasitism during the decline. And again, there was a model developed by Tanapana and others, and they tried to show whether parasitism itself would be enough to create these dynamics. And they were unable to confirm the role of parasitism alone. To my knowledge, and I may be wrong on this, but to my knowledge, nobody has attempted to modify those models and include a fecundity factor. Next, I want to talk about my favorite system, and that's the Western tent caterpillar. We see here on the left the type of family group of caterpillars that you get. Underneath that, they're sitting on their silk tent. They arise from a single egg mass and stay together as larvae, go off and feed and come back to the tent. In this part of the world, they use these tents to bask in the sun on cool spring days, or to hide under the tent when it's raining, or when they're molting, they go into the tent. This is the late instar caterpillar here in the middle. When they reach the late fourth instar and go on to the fifth instar, they begin moving around. You cannot support a whole group of these late instars at one site, so they move around the trees. Over here on the right, we see trees that have a number of tents on them, and you can see the sort of brown that indicates areas of defoliation. Well, Bill Wellington in the 50s and early 60s started working on the tent caterpillars in British Columbia. We can see that they are native to North America, although I see that their distribution does tip down into Mexico. Excuse me. More recently, my colleague Jenny Corey and I have been following these populations in Western Canada. And this is the sort of pattern we see. We've been looking at them in island populations here in the red dot or red star here on Galiano Island. The green star is Mandardi Island, and over here the blue star is Saturna. These islands, the Southern Gulf Islands, they're called, occur between Vancouver and Victoria, and they provide a nice study system where we have rather distinct populations that we can follow. We see that they're quite well synchronized over time, although the blue population here does seem to reach a peak a year before the other populations. And this is what it looks like when relatively high density here. You can see the size of this tent. It's very large. As I mentioned, sometimes you also get caterpillars hiding underneath the tent if it's too bright and sunny and hot for them. So the tent is really a mechanism for them to regulate their temperatures. When it's too cold, they sit on the top and bask in the sun. If it gets too hot, they can go underneath and use that for shade. This middle picture shows us what can happen at a very high density. This would be a picture taken in June, which of course is our late spring. All these red arrows are pointing to tents, and you can see that there are essentially no green leaves left on these trees. If you could see down here on this road, you would see caterpillars crawling across the road at high densities, going and looking for a place to pupate. If you came back to this site 2 months later, you would find that the trees had leaves again. So this is part of this system with these deciduous trees, that the caterpillars can totally defoliate them, but it's rare that they kill the trees. Usually the trees are able to recover later in the summer. They have enough time to come out and leaf again and build up resources. Here's another picture of defoliation, and you can see the tents again. So what we've been looking at is the number of tents, which is equivalent to the number of families, because the females lay their eggs in a single egg mass, as shown here. This shiny thing at the bottom behind the moth is really an egg mass that she has just laid. She would have laid that in probably middle to late June. The eggs very rapidly develop to very early instars, and then they go through the winter. They just sit there in an innate form and wait for spring to come. The next picture here is an egg mass from which the larvae have emerged, and you can see these tiny holes is where they have come out. And this provides a really pretty unique opportunity for us to measure the fecundity of the moths, because the moth only lays her eggs in a single batch. They stay on the tree. We can go out and find them and collect them, and then do the various tedious thing of counting the number of eggs. Egg masses tend to be somewhere between 150 and 300 larvae coming out of each egg mass. Over here, we have another picture of caterpillars not looking quite so happy. This caterpillar on the right has died of a virus that the caterpillars get. It's a nucleopolyhedral virus. And this caterpillar over here in the far right has been parasitized. You can tell they become sort of hard and torpedo shaped. So these are the two mortality factors then that are common in the population. Just a little bit more about this nucleopolyhedral virus. Here we have a tent with a few caterpillars on it. And if you look at that closely, this is the same picture I just showed you. And this pink goo here is virus seeping out. What happens, the virus is actually able to control the cuticle of the larvae, and it causes it to become very thin, and the very sick caterpillar just sort of melts. If we take some of that and put it under an electron microscope, we see these little balls, which are sort of protein. And if we take some of those and put them under higher power an electron microscope, we see this protein matrix and then these virons that are contained within it. These are spread on substrates. Probably they could get spread around by caterpillars walking through the goo. Rains come and it gets spread around to other leaves. And even when it dries out, the wind can blow it around, so that another caterpillar comes along and eats one of these little polyhedra, we're calling them, and the virus is released and infection begins. So some of the relevant relationships with the population regulation of the tent caterpillars is, first of all, fecundity is related to infection. Fecundity is also related to the density of the population, and the infection level is related to the density of the population. And I'll show you these relationships. Okay, here we have the percent families infected by virus. We collect from the families, we collect 10 larvae, we bring them back and we determine whether they have virus or whether they're parasitized. And then we have here the fecundity, the average fecundity of the population. So when the density is, when the level of infection is high, the fecundity tends to be lower. Now we found in earlier studies, a student of mine, Lauren Rothman, and I did this work that showed that larvae that are able to survive the infection, that is, maybe they were infected very late and they're about to go off and pupate, then they will still be able to get through and produce moths, but they will have a lower fecundity. So there's this sublethal effect of the virus that reduces the fecundity of the moths. Here we have the relation of the fecundity to the population density. The green is the average fecundity for the populations, and you can see as the population increases, the fecundity is high, and then it begins to go down as populations get crowded. It's relatively low during the decline, and then will increase again associated with the next peak. So again, we have an association between fecundity and population density in these cyclic caterpillars. And this is shown here in this relationship on the x-axis, the number of tenths, so the density of the population, and on the y-axis is the fecundity, the average fecundity of these in these different populations at these different densities. Then finally, infection is related to population density in a curvilinear way. So again, on the x-axis, we have the number of tenths, and on the y-axis, the percent of females infected the next year. And you can see that the number of tenths is related to the infection in the next year. So there's a carryover here of this virus being spread around through the population and overwintering, and then the next year infection will be high. Now, if we look at the relationship of parasitism and infection to the population density, we see this picture. First of all, the population density in the black line going up and down. In the red, we have parasitism. So this is the percent of caterpillars, percent of families that had caterpillars that were parasitized. It is higher here at low density, it's higher here at low density, and it's higher here at low density. So this is not causing the decline, but parasitism comes in after the population decline. Here's the infection level, and we see it's high when the population is high, and it drops down, and again, the next cycle, infection gets very high, and it drops down. It appears that parasitism can only be high when infection is very low. So these two factors then appear to be driving the dynamics of the cycles in association with changes in fecundity. Now, why is that trade-off between the virus and the parasite? Well, we did an experiment once in which we put out family groups at different times in May, and we actually added infected larvae to them so that they were infected. And in the early part of May, we found a lot of virus infection and no parasitism. But later in May, then the parasites come in and they begin to have an effect. If you have very high infection in populations, there are going to be few caterpillars around for the parasites to attack. So viral infection is higher in early families and less likely to be parasitized then. So there's this trade-off between viral infection and parasites. Now, what influences the rate of population increase in these populations? Well, here's a structural equation model done for us by Adam Pepe. And what it shows is that although parasitism occurs, it does not have an impact on the rate of population increase. So it is related to the density of the host population, both in that year and the year before. but no further impact on reproductive rate. What does affect the reproductive rate is the density in year T-1, the fecundity in year T-1, and the infection in year T here. So the red lines indicate a negative impact on the rate of reproduction. So that's the infection and the density in year T-1. And fecundity in T-1 is a positive effect on reproduction. So that summarizes it here. That just colored in here are the negative interactions, and the blue is the positive interaction on the reproductive rate. So my colleague's friend, or I mean student and friend, Paul McDonald, put together this model in which we have a component for infection based on that relationship I showed you previously, one for fecundity, again, based on our observed relationship. And we calculated then the reproductive rate here, the rate of population increase, and fed that into a model that looked at the density from one year to the next. And what we found was that if you had infection, fecundity in the model, you get quite a good fit here of the model dynamics in blue and the observed dynamics in red. And the way we did this, we used information from two populations to parameterize the model to predict what happened in a third population. And we were pretty pleased with the relationships here. If we took out the fecundity component of this population, we found that the cycles did not continue. So we see here the modeled dynamics. It begins with a cycle and then a little bit less of a cycle and less, and eventually it just stabilizes. So the take home is that without a fecundity component in the model to keep the density down for a generation or two, the populations do not cycle. Variation in fecundity is necessary for cycles. And again, this is by Paul McDonald's model. So to summarize the population dynamics of the tent caterpillars, they are driven by reduced fecundity at high density, high parasitism during the decline, and the model shows that changes in fecundity and viral infection can reproduce cyclic dynamics. And we feel that this sort of a model, although it's quite a simple model, but it still allows us to make this comparison between, is the parasitism, which sometimes can be very high, is that sufficient to produce cyclic dynamics, or do we need something else? And that's the fecundity. Again, this goes back to some of the earlier lectures that we've had about the cycles in the mammals, that it's not just mortality, but also it's reproduction that must be taken into account for cyclic dynamics. Something else that we have maybe been less good at quantifying, but it certainly has had a major impact on us trying to do lab populations, is that in general, the survival of the tent caterpillars is poor in those declining populations. We found that it's very difficult to do laboratory survivals. The survival was approximately 20% in one year that I looked at, in which we were trying to rear them in the lab. And again, this goes back to the snowshoe hares, that you bring them into the lab from those declining or low population densities, and they don't do as well as they do when they're increasing or high density. So something about the quality is changing. And again, talking to colleagues who work on other species of forest tent caterpillars, they have the same sort of experience. They tried to do some experiments in the lab, and they just had such poor survival, they had to give up. And of course, this can be devastating if it's a graduate student thesis involved. So reduced quality of the caterpillars as populations decline. want to mention briefly here another cyclic species that plays a major role in, particularly in Eastern North America, and that's the Spruce Budworm. Studies of Spruce Budworm really began to take off back in the 1940s, and between then and now, a tremendous amount of excellent ecology has gone on to study these populations and the population cycle. So this is a trend. This is based on measures of defoliation of the forest in Quebec, that was, and we see how the populations go up. And in this case, they stay high for a number of years before they decline, and then they turn around and come back up again, and again, a period of high density. Well, what's happening in this period of high density is it's initiated by high larval survival and moth fecundity, and then this period of defoliation, or bird predation is high here. There's larval and moth dispersal from the populations. Then fecundity goes down again, as it does with the other species. In general, survival of the larvae is poor. And then you get density-related parasites coming in here. Tree death and change in the forest indicated down here by you get the coniferous trees killed off. You get a period where you get some other tree species coming in, some deciduous trees. And then it takes a while for the forest to begin to recover, and then the population of the moths begin to increase again. During this period of time, really low density, there's been some experiments done that show that they get so low in density that it's difficult for the males and females to find each other to mate even. So there's sort of that Lee effect that keeps driving the population down. Even lower forest recovers and the population recovers. Putting this all together, then we have sort of this picture here where you do get in these species feeding damage to the trees. For the Western tent caterpillar, you get increased infection, and there are other species that get similar infections like this, the tussock moths and the gypsy moth, for example, both also get that same viral, different species or different types of the virus, but still viral infection. Fecundity is reduced, larval survival is reduced, and parasitism increases as the population goes down. Also, perhaps there is increased dispersal associated with those high density populations. I'll show you some pictures soon. This reduces the reproductive rate and the density goes down. And then there is a period in which the fecundity increases again because the density is down, there's no defoliation, no food limitation, no competition. And so the surviving larvae do very well, and they produce large moths, and those moths have high fecundity. Survival overall improves, they're good quality. Infection is low to 0, and then there is reduced parasitism. So this is pretty typical then of all these species. But I want to talk a little bit about this dispersal component. Often dispersal is not very well studied in population dynamics. And yet I think it is very important. Now this is one type of dispersal, and this is based on the autumnal and winter moth complex in Europe. This is work by Tenau and colleagues. And what they did was put together this picture of outbreaks over a number of years. And this might be a little bit complex, but it's looking at what starts out here in the eastern part of Europe. And here we have years on the y-axis, and this is distance from the ocean on this axis. And you see that you begin getting outbreaks down here in 1950, let's say, and then over the next 10 years, the outbreaks seem to spread to the west. The next period of outbreaks, again, it starts in the east and continues to the west, and so forth for these different periods of outbreaks of these populations. Now, I have to say that not everybody agrees with this, but I think it makes good sense that you would have population spread from a center where it begins, and by the time these populations are at outbreak, well, I shouldn't say from here, by the time these populations are at outbreak, these populations are ready for the next cyclic outbreak as well. So there does seem to be a pattern maybe of spread, widespread dispersal in these populations. As I say, not everybody agrees with that, but it makes sense to me. Here's another picture. This has to do with the spruce budworm outbreaks. And one of the things about the spruce budworm is you get these flights of moths at peak density. So all of these are spruce budworm moths that have flown in the night before. Hundreds of thousands of moths. It has to do with the weather conditions. Interestingly, it seems like when it rains, you get these flights occurring. So how much might that influence populations? Well, if you look at the spread of spruce budworm across North America, and when did they outbreak in the last period of outbreaks? Well, in Northwest Territories, the outbreak was 2002, 2003. I mean, it lasted longer than that, but that's sort of where it started. In 2006, 2007, it was recorded outbreaking populations in Ontario, and 2014, outbreaking populations in Quebec, and both in southern Quebec. And another phenomenon that's happening here is there's more populations spreading in northern Quebec in the boreal forest. So all this blue is the distribution of the eastern spruce budworm. And it has now spread on to New Brunswick and even Newfoundland. So there does, again, seem to be this geographic trend, and maybe a bit similar to the snowshoe hares that have synchronous populations across a pretty big area of Canada, but not all the way to the east. Well, these ones seem to be maybe spreading all the way across. Similar thing for the large bud moth. There seem to be waves of populations from population centers, and then they spread out down the mountain to lower populations. So we've looked at long distance movement that indicates that there is dispersal in these forest Lepidoptera, but I wanted to use another indicator that dispersal could be occurring frequently, and that is the synchrony amongst these populations. So here we have some information on the Western tent caterpillars, 3 different island populations. And we see that their cycles are very synchronous over time. We hypothesized that if this synchrony was associated with dispersal, then populations should be genetically similar. And we used microsatellite markers to look at this. Here we have information from populations during the one outbreak in 2003, the next one in 2011. And here we have different populations: Galliano, Mandardi, Pender, Saturna, and Westum. And although we see that there's considerable variation amongst individuals, these bands indicate the composition of the genetic makeup of individuals. So the amount of the yellow indicates certain markers and green indicates other markers. We've seen that there's a lot of variation here, but there's nothing to differentiate these populations either geographically or temporally between the outbreaks. So no genetic differentiation among populations or between outbreaks. We interpret this to indicate that there is considerable dispersal occurring. This lack of genetic differentiation among populations is indicated by their low FST value. And we searched the literature to see if other cyclic populations also showed this type of a lack of differentiation amongst populations. And we found that there seems to be this general pattern. Winter moth, which the females are wingless, but the larvae do this ballooning behavior to move around, they too had lack of genetic differentiation. Similarly, with the large bud moth, the spruce budworm, pine processionary moth. And we were surprised to find that some of the cyclic mammals also showed this lack of genetic differentiation. Studies were done on the water vole and the snowshoe hare. And we also looked at migratory locusts. So we knew that they move quite distances, and they too show this lack of genetic differentiation. So we think that movement is a characteristic of cyclic populations that is very important in maintaining the dynamics amongst areas and over time. Now, another question is it ever possible to stop cyclic dynamics? And with these forest lepidopters, which sometimes are considered to be pests, there's been a number of spray programs that have been initiated to try to stop the population outbreaks. I have some data here from a sawfly in eastern Canada. And what we see is 3 different years in which the populations were sprayed at the time of this dashed line here. And they were sprayed with a nuclear polyhedral virus that we have talked about as being an important factor in the Western tent caterpillar population dynamics. So here the populations were sprayed, and there's little difference between the control and sprayed populations. The next year, we see the sprayed populations that are slightly lower than the control populations. By the next year, the population, this is peak population now, they're a bit higher than they were previously, and both the sprayed and the control populations decline over that summer. The next year, we have here the sprayed population slightly lower than the control populations, but doing quite well during this summer. And finally, during the population decline, the regional decline, the populations do, both the controls and the sprayed decline over that summer, although this control population stayed a bit higher. And the next year, populations of all, both sprayed and control, are lower. This sprayed population, in fact, though, was a little bit higher than the others. So what this seems to indicate, and as I said, there are many other examples using other insecticide sprays or other microbial sprays that indicate you can protect the foliage for a year by spraying, but the only time the cycle stops is during the decline. So if regionally the populations are going down, then the sprayed and the controlled populations decline in synchrony. Now there's some other examples we can use to look at this question. Large bud moth, for example, which has been shown by tree rings to have gone through these cyclic dynamics for several thousand years, actually. But recent warming caused an elevational shift in the large bud moth populations, and they moved up the mountains and eventually ran out of suitable habitat. And this led to a dampening of the cyclic dynamics. However, in other parts of Europe, large bud moth did continue to cycle. Western tent caterpillar, we attempted to stop a cycle by cropping an island population. Our idea was that if we kept it low, then it would have all the characteristics that would allow it to increase once we stopped the cropping. But that did not occur. The population kept going up from one year to the next, and the decline was in synchrony with surrounding populations. So our interpretation of this is, again, even though this is an island population, we weren't able to stop the immigration into the population, and therefore the population just followed what was happening around it and declined. Gypsy moth, introduced to North America, had another interesting situation where a fungal disease suddenly appeared. This is a gypsy moth larva, and this is areas in northwestern US that are defoliated by these gypsy moths. And these are data that go a long time from 1924 through, I think, this 2013-ish year. And this is measured by the hectares defoliated on this scale. So when there's a lot of defoliation, you have high gypsy moth. Now you can imagine that the estimates of defoliation have gotten better and better as more airplanes were used and that type of thing. But it wasn't until about 1980 that there was a massive outbreak of the gypsy moth in Eastern North America, followed by a decline, and then another outbreak in the 90s and a decline. And then since then, the populations have stayed relatively low. Again, this now is not on a log scale, so these little blips are sort of real measures and say less about population increase than actual numbers. So what happened in 1990? Well, there was a fungus that had been introduced years and years before, but never seen in populations until 1990, when all of a sudden it was very common in these northeastern US populations. The population declined both because of infection with this fungus, entomophaga, and as well as these populations also have the nuclear polyhedral virus. So there's a lot of virus and a lot of fungus, which now is adding new mortality to these populations. And after that, the populations stayed low. And this work was done primarily by Ann Hayek, who's at Cornell University, very excellent insect pathologist and entomologist. Now we can see what happens if you get rid of the fungus, which occurs in dry years and when there is no fungus, and the gypsy moth populations took off again here in the mid 2000s. So that is sort of a natural experiment maybe that indicates the impact of the fungus on the populations. I don't have the data since then and what has happened, but when the rains come back, the fungus should be back, and that should cause the population to decline. So the summary then is that there are similarities between fluctuations of species and in what causes the declines. Mortality is either parasitoids or fungal disease, or viral disease mostly, sometimes fungal disease. And also reduced size and fecundity at high density. This seems to be a common characteristic. Dispersal waves of spread and high variation, but similarity amongst populations. Some questions: why don't populations cycle in the south? Could it be related to generalist versus specialist parasitoids? Is it related to the phenology between the insects and their host plants, or maybe even the host plants change? Or maybe there are cycles in more southern areas, but they're just not recorded. So this is an area that we really don't know a whole bunch about. What determines variation in the peak densities amongst these outbreaks? Again, this is true of, I think, the snowshoe hares and the voles, and certainly the forest tent caterpillars. And will climate warming have an influence on these populations? I think this is a question that still we don't know an answer to. Now I want to do this comparison to a non-cyclic species, the Monarch butterfly, just here at the end of this talk. As you well know, probably, they feed on milkweeds, which are herbaceous plants. They migrate long distances each year. Unlike the forest caterpillars, they are more likely to be spreading their eggs widely from one patch of milkweed to another. And they have 4 generations a year, which gives them a great potential for population increase over the breeding season. Now, one of the reasons I was so interested in this topic is because when I was a graduate student back in the 60s, I was involved with a protozoan expert in actually identifying this species of protozoan parasite that the monarchs have, because I was trying to rear caterpillars in the lab, and all of a sudden they began getting this disease, and I wanted to find out more about it. Found out that nobody had identified it before, so did a lot of basic work, and then it led to getting this almost impronounceable name. So the system is here where you have butterflies that are infected. They have these spores on them. The spores are spread to the leaves. Where they're eaten by caterpillars, then the parasite develops in the caterpillar. It rare, unless it's very high density, it doesn't kill the caterpillar, but then the moths that come out are smaller, have lower fecundity, and sometimes deformed wings. And whoops, just to mention that like the cyclic species, the monarch butterflies also have low genetic variability, and that could well be associated with their migration each year up to the northern regions and then back down. So low genetic variability is, or amongst populations anyway, is related to migratory behavior. Now here are some population data. And there are two ways to estimate the populations of monarchs. As again, you're probably well aware that the size of the overwintering population can be estimated. And we see here that it was much higher early on and has been going down very low here and then has come up a little bit. Okay, so that's what's going to the south and overwintering, but what's happening in the north? How much reproduction is occurring up there? Now this is a paper in which it's based on doing visual sightings of moths flying by. And this indicates maybe a little bit of a different pattern in that there's no trend for decline here. There's a lot of variation. I think in 2004, the population from the north was very low, but then it recovered over the next couple of years and has bounced around. Down here, I have plotted from Altheiser's work some information on the spores. And in these migratory populations, they tend to be very low and not density dependent. This is another data set on those northern populations and visual sightings. And again, a lot of variation over time in terms of the numbers, but not the sort of drastic decline. This is from Agarwal and Inamin's work in 2018. They summarized this, and we see populations in the Midwest in red, in the Northeast in yellow, and they vary considerably from year to year without a particular trend. Down here in 2014 to 17 is a comparison of populations in the Midwest, the Northeast, and in Mexico. We see there's no strong connection like in 2015 here. High densities coming down from the north in the Midwest, but not so high from the Northeast, and the overwintering population was relatively large. So there seems to be certainly some disconnect between what's happening in the breeding populations and what's happening in the overwintering populations. In 2014 was a particularly bad year overall. I guess part of the moral is that if you have bad conditions in the north, that can really have an impact if there's also bad conditions happening in the southern population. Here is another structural equation model that looked at the relationship between glyphosate, which is thought to really reduce, this is a herbicide spray that's used widely in the Corn Belt of the US, and it eliminates a lot of the milkweed along roadsides or certainly in the fields. So you have high glyphosate use is a negative impact on milkweed resource, and the amount of milkweed you have is very important to the number of butterflies coming into the overwintering area. And another factor is August temperature here, because high temperatures in August can be very stressful to the migrating butterflies and reduce their density. So monarch, this is according to Flarcott study, that the monarch abundance was more than 4 times more sensitive to perturbations of the vital rates on the breeding grounds than on the wintering grounds. Important factors are summer food plant availability and local weather. The winter forest protection and these extreme weather events, and whether that's going to be associated with climate change or not, will is perhaps anyone's guess, but these hurricanes going through. And then migration can be a detrimental state for the butterflies coming south if it's too hot in August, and probably also poor conditions in the spring can have a major effect on how many moths, the butterflies are getting to the north to breed. One little quick thing here, and this is the total monarchs reported in the West. So this is the Western population. And we can see that the recent droughts in California have been associated with extremely low sightings of monarchs. So again, a weather factor having a strong impact on the population. Now there is some density dependent crowding which occurs on the food plants. This again, a Flockhart study. If it gets more than 5 eggs per plant, the pupil size will be reduced, and associated with that will be a reduction in fecundity. And weather being a density independent factor, storms at overwinter sites, temperature during migration, and drought are very strong impacts on populations. Now, a question I've had is, I wonder what the availability of host plants was way back before humans got there. It does seem to be related to human activity now, but maybe there has been a period of time in which this disturbance that humans caused created a lot more habitat for milkweed. We have roadside weeds, the agriculture opens things up. So I'd like to know more about what was the historic densities of monarchs. Conservation is very complex, and one thinks of a comparison to the Stan Bouten's talk on the caribou populations and the complexity of trying to conserve them. So again, a huge problem and very difficult to solve. Okay, quick comparison. Cyclic species are capital breeders with short-lived adults, lay eggs in masses, strong density-dependent mortality. Their host plants tend to be trees, very large. They disperse and don't migrate, although they do move across. And monarchs, income breeders, butterflies, long-lived and feed. The eggs are spread amongst plants, strong impact of the environment and food availability, and they migrate. Forest considered caterpillars are often considered pests, and monarchs are a conservation concern. So they're two extremes in how we look at them. For monarchs, protecting the food in the north, protecting the overwintering sites, and what the impact of climate change will be is to be determined. I'd like to end by thanking those many, and there's just a few people I've named here, but there's so many people that have worked on these cyclic species over the years. And of course, underneath in that pyramid are the numerous graduate students and undergraduate students who have done so much field work, and particularly I am appreciative of those that have helped me with the tent caterpillars. And thanks for your interest.
Thank you very much, Judy. Great talk and great caterpillar. I'd like to know more about those colors and I will ask you. But before we go into the questions, there is a lot of greetings and congratulations from many states in Mexico, from the north of Baja California to the south in Campeche and Yucatan, and even someone showed up from southern Panama, and that's great. And it was an amazing talk that you presented a series of studies where the data go for decades, and many excellent ecologists, and also the way you presented the different aspects, first with mortality, then the issue of fecundity, then dispersal, genetics, really incredible. The first question I'm going to raise, which comes to my mind, is why 10 years? Why 10 years? These insects recover fast, unlike mammals, where mammals have these lows. You don't have these lows here. They recover fast, but they still go 10 years. When you have such a number, I guess people always think about something external to do with weather or something like that. But why 10 years?
Well, I think it has a lot to do with their the reproduction rate, and it does seem to be typical of these cycles that the increase phase is a bit slower than the decline phase. So when those mortality agents get in there, it can knock them out pretty quickly, but still over a couple of generations. And then if you have the fecundity thing, that could contribute to slowing down the increase the next phase. Now, even I have fallen into the trap of thinking, oh, well, sunspots are 10, 11 years. Maybe that's a factor. And it isn't, because there has been a study that looked at the synchrony between the caterpillars and these sunspot cycles. And because sunspot cycles are slightly longer, you can get periods of time, which can be relatively long, 30 years or something, and they seem to be synchronized, but then they begin going out of phase. So from way back, that was always a popular idea. Oh, it's got to do, and it could do with weather, because sunspot cycles do influence the weather, but it doesn't hold up. It's an attractive idea. And of course, then other patterns, cyclic patterns, have been looked at, but really, I think it's all internal to what the reproductive rate is and what those mortality agents are.
Okay. And in talking about fecundity and this increased rate, it looks, well, you showed it's important. And what is the relation? I mean, what I see from the talk is fecundity increases with the size of the caterpillars, but also, what is the relation between fecundity and the food quality? Is there a relationship there? Maybe there is also a relationship of fecundity with the parasitism, but what is the relationship, or is there a relationship with food quality?
Yes, there is, and we've done some experiments in which we have fed caterpillars leaves from branches that had other caterpillars feeding on them. And so that, you know, the leaves are maybe partially eaten and that sort of thing. Well, it's poor quality food. And those moths tend to be smaller. Also, parasites can have an influence on fecundity, because often they feed on the larvae. So they will sting the larvae and then they will feed on hemolymph, and the larva can still live, but it will now be reduced in size and fecundity. So there can be that association. And then a sublethal virus. People have really worked to try to show that induced plant effects drive these population cycles. And even the most strong proponents of this idea have had to say, no, that doesn't work. So there are short-term impacts on the food quality and the food quantity, which I think is also a major factor, because you do get defoliation. But the long-term delayed effects don't seem to be driving these population cycles.
It looks, it made me think that these caterpillars are like three rings. You have small ones that come out from hard times, and then big ones that come out of good times. But
well, it's fascinating that the tree rings have contributed to a number of studies of these caterpillars, because the caterpillars leave their signal on the trees. If defoliation occurs, as you can imagine, then the growth of the trees is very reduced. And some people have gone back several thousand years to look, particularly with the Spruce Budworm, there's work, with the larch bud moth, there's work. And so you can take these cycles and go way back in time before any of us were alive, and show that it has been a pattern that keeps reoccurring.
That's great. I wish we had the same for the monarch butterfly, because I'm also, as you, I also wonder about what the densities of the monarch butterfly were before, since like they were discovered, or the migration was discovered in 1975, which is a bit after the prohibition of DDT in the United States. So, plus also because it's a butterfly that lives off plants that live on crops, then for sure it has increased. But I I have here other questions. I'm not going to hog all the questions. One question from Carolina Horta. She asks, what are the principal causes of the viral infections?
What are the causes?
The main causes when you talk about these viruses.
Yes, that's pretty fascinating as well, because when we look at the low density caterpillars, we don't pick up any infection. And then all of a sudden, when the densities get high and they start being crowded, then infection occurs. And of course, then it can spread very rapidly with the high density populations. We think that perhaps there are what are called innate forms of the virus that are not actively replicating and killing the caterpillars, but you can pick it up with molecular techniques, that there are these latent viruses that you can find. And but what we have not seen is, well, what if there is a latent virus, what makes it change from latent to active forms that now kill the caterpillars? And that's a big question. It could be that it's just hiding away in the environment, and then when you get lots of caterpillars, they start picking it up. And a lot of people think that that's the case. It could be in the soil, it could be on the trees. I think they're carrying some of the virus along with them. They get stressed, and then boom.
That's interesting. She also asked if these cyclic phenomena have been recorded in diurnal butterflies, or I guess most of the examples that you show are moths.
Yeah.
She wants to know if there is cyclic dynamics in butterflies, in diurnal butterflies.
Not that I know of, and I would like to know much more about that. And as I said, that I don't know if populations in more southern areas or more tropical areas just haven't been studied the same way, or if they're so much more diverse, the number of species you have, that any of them are not going to reach that high densities. That's for the next generations of insect ecologists.
Yeah, that's one of the things that I was surprised, like these systems that you presented are driven by a couple of factors, 2-3 factors, no? The parasites, the virus, and in some cases the fungi, and the so it's not it's one thing that there is many causes of mortality, but in these systems it looks like 1, 2, or 3 are really determinant of driving the cycle.
Yes, and of course there's lots of things that we don't study. For example, I saw a recent paper, there are these small RNA viruses that caterpillars have sometimes, and they tend to be more impacts on the sort of size and fecundity than the mortality. But yeah, there's many things that we don't know about other mortality agents. But as I said, we do know that it's jolly hard to do experiments in declining populations because they die.
Yeah. There is one question from Jose Luis Aguilar Rodriguez, which I would like to link to another one. He asked that if there has been an influence on these cyclic populations with the use of pesticides, when the pesticides are sprayed in the forests. But with that question, I also have one that is all this information. I mean, you show some of these are pests, or all of them. I don't know. I don't know if the one on birch it's a pest too in Europe. But the question is, how is that this information on understanding this cycle used to apply to pest control?
Yeah, well, I tried to show a little bit about that, okay, pest control, you're trying to protect the foliage in an area, and you can't spray everything. So I think the spruce budworm maybe is the best example of that situation, where it's very widespread through the boreal forest. And you can spray areas that are high value forests and try to keep the caterpillars down. You have to spray every year in order to do this. But then once the population declines, then you're sort of home free for another number of years. So yeah, I don't know that it tells us much about what we could do with non-cyclic species, but outbreak species are going to be the pests, and outbreak species on crops or on trees are going to be pests. You can spray them, you get resistance, and then you're back to where you were before. But I think that many of these forest caterpillars live in areas where people don't like their defoliation very much, maybe, but it's not crucial. Like with the tent caterpillars, people get really annoyed about them crawling along the roads and crawling up their houses, which they do when they're defoliated everything. But they're not a serious pest. Apple trees, yes, but then you can spray your apple trees and protect them for those couple of years.
Okay, and just going towards the end, you talk about the monarch butterflies, and it's been interesting because there is some papers that predicted that with the global warming, the monarch butterflies were going to go higher and higher up in the mountain because of the temperature changes. But what has happened in the last 2 years is that they have returned to the north 1 month earlier. So it's quite surprising. They used to leave mid-March. They used to go mid-March back to the north, but now they are going mid-February. And one question I have is that every year, as you know, there is an estimation of the area that they are using, and the reporters, the news reporters, are always very attempt to this. This year they went down 25%, and they always ask, are they going to go extinct? Are they what's happening? But to me, it's not surprising that they go 25%. I mean, it would be surprising if we had the same number every year.
Yeah, yeah.
These are insects, and they are these 4 or 5 generations, and so when should we be surprised in changes of density?
Never. But I do think that, maybe this can be beneficial, because if you have a decrease, that hits the newspapers. That's in Canada, that's in the US. Monarch populations down 25% in the wind. And I think that brings people on board. And well, as well as anybody, the number of citizen scientists that are out there putting little tags on butterflies, or counting them, or rearing them to release, planting their milkweed. And of course, the story there is you want to be careful about the species of milkweed, because the Asclepias curassatica, which everybody loves, is not a good food source for them. And then the other factor is that if you get too many populations that don't migrate anymore, as you do in Florida, then this protozoan parasite does build up and has more of an impact. So got to be careful about what types of interactions there are with humans trying to do good. Yeah.
Well, so we should keep them.
Nope, nothing's going to be stable.
We should keep them in the news.
What's that?
We should.
Yes, absolutely. I think people love them. It's how many things you see that the monarch is a symbol of happiness, butterflies.
Yeah. Well, thank you very much, Judy. It's been a great afternoon. Thank you for your time and for being here with us. And we will continue next Thursday with one more session with Dr. Sinclair, talking about the dynamics in the Serengeti ecosystem. Thank you, Judy.
Thank you, Carlos. Bye-bye.
Bye.